Hi. So thanks for inviting me for the grand rounds. I'm a joint faculty between otolaryngology and biomedical engineering. I know many of you work very closely with colleagues from otolaryngology. And I'll tell you a little bit about one part of the research that we do in my lab, looking at hearing preservation and using therapeutic hypothermia. So just some conflicts of interest that I want to declare at the beginning. And there are two parts of my lab. You could say that I work with temperature, really. So the top part works with hot stuff. So we figure out how we can use photothermal energy to stimulate neurons directly and think about how this could be applied in the ear and elsewhere for example, neuroprosthetic applications. So this is sort of similar to what you guys might know about optogenetics, where you use opsins and transfect cells and then use light to manipulate activity of those cells. But this is a direct photothermal application. But I'm not going to talk about that today. I'm going to talk about the cool side, mainly because I know that hypothermia has a significant history in neurological surgery and preservation of function, right? So I'll tell you couple of interesting applications that we're developing in my lab, mostly for hearing preservation. I'll talk about cochlear implants and also a noise-induced model that we have. So hypothermia, I don't have to tell this group about all the literature and the history of using hypothermia, particularly mild hypothermia as it is used for neuroprotection. There is a significant data from animals, significant clinical studies going on. Many of them have actually failed, right? Like if you look at the clinical trials for hypothermia, there's not a lot of positive results. But yet, this has a lot of promise, and one way that I particularly think that this has significant promise is through using a localized approach. So if you can deliver something very locally to an organ, especially something that's as sensitive to temperature as the cochlear or the vestibular system, maybe there are applications. And I'll talk about two preclinical models that we have in my lab. One is the cochlear implant, and one is a noise-induced hearing loss. So again, the auditory vestibular system, I'm pretty sure that you guys know the system very well. Many of my colleagues a lot of times will ask me, don't you get bored of working on this such a small organ? But I just think that this system is actually really, really beautiful, right? It existed, if you look at actually fossil records, the vestibular system was actually, it's found in 400 million year old samples, right? So it was there, and since then it has really refined but not really changed significantly. And a similar thing for the auditory system or the cochlea. I'm not going to talk about the vestibular system today. That's probably for some other day, but I'll talk about the cochlea. Again, the structure and the function, I think you guys probably are very well aware of it. If you're not, let me know. I'm happy to elaborate. But you can kind of unfold this structure, this spiral of the cochlea that you have, and you can think about that, across that entire length, you have different frequencies being represented, right? So you have the base where you have the very high frequencies, and the apex where you have the low frequencies being represented. And that's because of the mechanics. That's because of the thickness of the basilar membrane, the fluid vibrations that come in through the oval window, through the stapes, and vibrate the basilar membrane, producing depolarization in the hair cells and the neurons, transmitting information, right? And as we age, what we end up losing is most of the high frequency, right? We still keep some of the low frequencies. All right, let's go next. And I just wanted to kind of show this little video of an outer hair cell. So this is an outer hair cell from a guinea pig that has a patch clamp electrode coming in. And you'll see what, when John Ashmore from UCL converts music. Is it playing? It was playing yesterday when we tried it. Anyways, one of the really beautiful things about these cells is that if you put electrical information into it, right? So if you put electrical energy into it, it'll change its size, right? And so this is what an amplifier. It acts like an amplifier in your ears, right? So the outer hair cells, there are 3 rows of outer hair cells. There are 3,000 inner hair cells, but about 9 or 10,000 outer hair cells, right? And what happens is when you put in energy into it, which is In a normal cochlea, it's a vibration of the basilar membrane that comes in, right? As the fluid moves, these outer hair cells, they'll elongate and shorten. They'll keep their volume, but they'll elongate and shorten. And what that does is that actually ends up by about 30 decibels amplifying the sound that we have. So as we age, when we lose these outer hair cells, right, what you lose is that ability to amplify the sound and hence hear the low frequency sounds or softer sounds, right? So then you need, like, for example, a hearing aid, which is essentially a loudspeaker, to make things louder. These cells also are extremely sensitive to damage. So maybe I think it's, so you can kind of see, like, how it vibrates, right? So there is actually a motor in the plasma membrane of these hair cells that's a piezoelectric. Right. So it's a piezoelectric sensor. So it takes electrical energy and converts it into mechanical, or takes mechanical energy and converts it into electrical, right? So it allows it to elongate like that. So it's a beautiful system, and it was just one of the things I wanted to show you guys. I had a couple other videos that just for time I'm not going to show. But one of the things that happens in a lot of patients is either through genetic mutations, mitochondrial deafness, meningitis, trauma, that you end up losing a lot of these hair cells. And along with that, a lot of times you'll also use inner hair cells. Now, the inner hair cells, outer hair cells are the amplifiers. The inner hair cells are the ones that send information to the brain, right? So if you lose those, you have no way of transmitting sound to your brain. So the way, and this is really an engineering marvel in some ways, and every day I'm surprised that these devices work so well, but these are cochlear implants, right? And you can see on the right side in the bottom is actually one of the cochlear implants where you can see the portion that goes inside the ear. So the tip, that little spiral tip that you see at the end, that's the one that's inserted into the cochlea. The surgeon will go in, open up the mastoid, approach the cochlea through a facial recess, look at where the round window is, which is your natural opening into the cochlea, and either insert the electrode through there or make a cochleostomy or a hole on the basal turn of the cochlea or the bone itself, and then insert the electrode, right? And then you have a speech processor that sits outside and a transmitter. So as the sound comes in, the microphone picks up what the sounds are, decomposes it into like all the different frequencies, transmit the information to the electrode then stimulates the remaining nerve that you have, right? And that's how, about 500,000 people around the world actually hear through these devices. So they've been really, really successful in giving hearing. One of the problems, as you can imagine, especially as surgeons, is once you go past the round window, you have no insight into where you're going, right? So you're kind of putting in this electrode by feel. A lot of the times, and yes, there are pre-curved electrodes and things like that you can get. But what happens, and you can see that in these micrographs shown up, micro CT. This is actually a human cochlea, and they took micrographs as the electrode was being inserted. And what you see in the center, that little spiral, that's where most of the sensory structures are, right? So you don't want to try and hit them. You want to get close to them. You want to put the electrode close to them so that your electrical energy required for stimulation is low, but you don't want to hit them. Because the moment you start hitting them, you're going to cause trauma. And you can see that right there in that A, you're actually kind of scraping that. In B and C, you're actually hitting that structure right away. So at that time, the surgeon usually pulls the electrode slightly back and then inserts it again, right? So this goes on for several minutes, and what happens during this time is that you're causing a lot of trauma to these sensory structures. This is actually from a colleague, Randy Water, who's now an emeritus professor here. He created this model of electrode-inserted trauma in an animal model, right? So he did guinea pig studies. And what he showed is that as you put in these electrodes, what you're doing is you're causing trauma, losing majority of the outer hair cells that might be remaining, right? And with that, you actually also end up losing inner hair cells. Now, recently, you guys may or may not be aware, but recently the FDA lowered the criteria for what it means to be legally deaf, and hence the criteria for who can get a cochlear implant. Okay? So if now you are someone who has a little bit of residual hearing, which is 70 decibels, right? So it's still something that your hearing aid won't benefit you, right? But the cochlear implant previously was not available to you because you needed to be 80 decibels or above. So now you have this spot where you have a little bit of residual hearing, meaning that you have some of the inner hair cells, maybe some of the outer hair cells preserved, And you want to do cochlear implants in these. So as you're doing this implant, what ends up happening is in majority of the cases, we end up losing the residual hearing just because of the trauma that's caused by the electrode, right? And we know that there are multiple different things that go on, like hair cell loss, there is fibrosis around the electrode, there is mechanical damage that we cause to the basilar membrane, et cetera. And so hearing preservation approaches are required, right? So especially to improve outcomes. And this is especially true if you think about even conventional electrodes. So somebody who is deaf, completely deaf, requires an electrode, and you inadvertently cause damage, trauma to these structures, you're actually also losing the neural fibers, right? So the very structures that you need to preserve to even transmit the electrical information are getting damaged. So there are ways in which we need to figure out how to keep these sensory structures from getting damaged from these electrodes. So I'm trained as an engineer, so one of the goals that I had coming into this was to think about, can we go beyond, pharmaceuticals, given that we know how expensive it is to make, get drugs into the market, but also how challenging it is to put drugs into something like this, a electrode like this, for example, and then create maybe an electrode that would elute drugs very controlled into a cochlea, which is, again, a very delicate structure. So we looked at the literature, working with Dalton Dietrich, we looked at the literature and said, hey, maybe, if we can figure out a way in which we can give local hypothermia to the cochlea during the surgery, would that help us, right? So we actually built a custom system, and I'll talk a little bit about that. And one of the goals is we don't want to modify the CI surgery anyway, right? You guys are surgeons, you know that the moment in your surgical space a new thing gets introduced, if it's hampering what you want to do, right, you're not going to use it. So what we want to do is we want to create a system that doesn't change the surgical approach that we have right now. And then we want to think about how we move into clinical translation. So here's a system that I put together in the lab. All it is, it's essentially a Peltier system. So I don't know if you guys know about Peltier systems, but if you take two metals and put them together, and if you pass electrical current through that, one will become cold, one will become hot, okay? And this is used in your cars, this is used in your refrigerators all the time. And the way you use these systems is, and the way that we use the system is, here's a Peltier element, we put a little fluid into this thing, there's a pump that circulates that fluid, and that fluid touches the cold side. The hot side is over here, and we take the heat away, okay? So we can cool the cold side. Through the cold side, we can cool the liquid, and the liquid is actually perfused through this little probe. Now, this little probe is shown over here. So it's a little metal probe at the tip, 5 millimeters long, 1.6 millimeters in diameter. It has a little microthermistor in there so we can measure what the temperature is. And all we do is we just circulate cold liquid through there. This little probe, is what we put right next to the cochlea. And through contact, through heat transfer, it ends up cooling the cochlea, right? And what we do is we created a little protocol where over 10 minutes we cool down the cochlea. We maintain it for about 40 minutes. Halfway through, we do the cochlear implant, and then we rewarm it up to 37 degrees again, right? So we created this protocol. We said, all right, we have something that's, maybe might work. Let's try it in animal models and see what happens. So we went ahead and, we got 3 different groups of animals, essentially 2 groups of animals, normothermic cochlear implants. So we implanted rats with cochlear implants, and we implanted rats with cochlear implants while giving them the hypothermia treatment, okay? And then we always use the contralateral cochlea for controls, right? And so what happens? So we can test the function of the cochlea in these rats by using auditory brainstem responses. So we can put, electrodes on our back of our ears, compare it to a reference electrode and a ground electrode on the neck or on the pinna, and when we give different tones, right, as the sound goes from the cochlea to the cochlear nerve, to the cochlear nucleus, to the inferior colliculus, and to the auditory cortex, you can pick out what those amplitudes of those different signals are. And those are called auditory brainwave responses. And what we can do is we can pick up one of those waves. So for example, the wave 1, which is as the sound energy, the electrical energy is traversing through the cochlear nerve, right? You can pick out that wave, and you can take the amplitude of that wave, and you look at at different frequencies. So if you give, for example, I gotta go back, okay. If you give one frequency, this is 16 kilohertz, you can look at what the threshold is, right? So what response do you get back out? This black line is the contralateral ear, right? So if it's flat, that means good, right? Softer sound, and it can hear throughout 28 days, right? The louder the sound, worse. That means that you're losing hair cells, you're losing neural structures, right? You're causing damage. The red curve right here, that's the normothermic rats. The blue curve right here, that's the hypothermia treated rats. And that was only treated one time at the time of surgery locally, right? And what you see, what you might appreciate, getting used to this thing. What you might appreciate is that even on first day after surgery, there is significant threshold shift towards protective side. And by day 28, it's pretty much similar to the contralateral ears, right? So you can preserve pretty much all of the hearing. And we can look at the entire cochlea, all the different frequencies starting from low frequencies. These are the most important frequencies for humans, right, to preserve, but also high frequencies because that allows us to kind of appreciate different things when we are thinking about hearing in restaurants or in background noises, for example. And what you see is across all of those, we can preserve residual hearing really, really nicely, right? This is data from 7 different animals in each group that we have, right? So we're kind of excited. We were excited about that, and we said, all right, that looks great. What does the structure look like, right? So we looked at the cochlea. These out here, these are your 3 outer hair cell rows. In here is the inner hair cell row, okay? And these are all cell nuclei. You can kind of see the inner hair cell nuclei right around here. This is a normothermic implanted cochlea at 28 days. You can see the amount of damage that the electrode causes, right? And this is a hypothermic preserved cochlea, right? So really, really nice preservation of structure, looks very similar to a control cochlea, matches the function that we saw. So again, things that are great. Engineers, we make, 3D diagrams, you love it, So what this is another set of experiments which was now done up till 3 months, so 84 days. All the different frequencies, so this is the cochlea sort of opened up, if you will. The red and the blue curves are again normothermic and the hypothermic cochlea, and you can see that by 28 days, 14 days, it's significant recovery, and by 84 days, I guess again pretty close to where we expect it to be. So we're kind of excited about that. Looked at quantifying what the hair cells do. So again, outer hair cells really nicely preserved with hypothermia. Inner hair cells don't really get damaged significantly, but I'll show you something very interesting in a little bit about what happens with these inner hair cells and the synapses that connect to it. And we wanted to show that anytime you're introducing something new, you want to show that it's safe, right? So what we did was we did 10 animals in which we only did hypothermia. And we looked at the hypothermic ear and then the contralateral ear. And there's no significant difference between either of them across the cochlea. So that means that hypothermia by itself is not damaging the hearing, right? So all good news until now. All the data that FDA would need if they're thinking about an IND or something like that, for example, to start a clinical trial, which we're actually hoping to start in Miami sometime next year. Scientists, we want to also learn about what the mechanisms are. So we did a whole genome transcriptome analysis, right? So these are control cochlea. This is a whole genome sequencing data, RNA sequencing data. So this is one sample in the column. These are control cochlea, so no cochlear implant surgery, no hypothermia. These are normothermic animals, so they received the cochlear implant. And these are the hypothermic cochlea receiving cochlear implants, right? And what you see is one is that normothermic implants, they look quite different, right? So there is a lot of gene changes happening between controls and normothermic. Hypothermia, on the other hand, seems like it's pretty white, right? So it's kind of suppressing everything, which is what we expect it to do. Hypothermia essentially suppresses metabolic activity and things like that. So it's kind of suppressing a lot of these different things. We can look at within using some of this gene data, we can run gene ontology analysis and look at what the biological processes that get affected. Again, things that we expect, oxidative stress. We expect that when you cause trauma, you're going to get oxidative stress, reactive oxygen species being produced, and that will cause DNA damage, for example. Chemokines, TNF-alpha, and all the processes related to that. Inflammation, which is essentially all related over here. Apoptosis, so caspases and things like that get activated. Hypothermia actually significantly reduces some of those. And then, of course, necroptosis, right? So programmed cell death. And we can keep looking at like, what these different pathways are. We can start analyzing them. We're doing now like, further analysis by using RT-PCR, Western blots, so to kind of look at like, protein analysis and think about are the right things happening with hypothermia? And all the indications until now are that they are. Again, like, you can kind of see like, caspases are down, significantly down with hypothermia, right? TNF alpha up till 14 days is down. So you can see that one-time application at 0 days affects gene changes until 14 days in these cochlea, right? So it's actually really interesting to me how one-time application of hypothermia continues to have significant effects like downstream. We can do like, immunohistochemistry, to look at like, MMPs and things like that. So just like we think about in the brain, the blood-brain barrier, right? And you don't want to do things that damage the blood-brain barrier. There is a very protective mechanism in the cochlea called the blood labyrinth barrier, right? So again, like, the blood vessels in the cochlea that might get affected, that might get pinched as you put this electrode through and cause like a reperfusion injury, which again, like, I think a lot of you might be aware of already. So we can start looking at some of those, and we see that they're also getting regulated by hypothermia. So again, all in all, like, good news so far. Being in a surgical department, I'm also interested in like, how do we actually move this into clinic, right? So we had a couple of residents and fellows working in the lab looking at human temporal bones. So they dissected out how they would do cochlear implant surgery in temporal bones. We put our little hypothermia probe through that facial recess that a surgeon created. This is the surgeon actually drilling furthermore of the facial recess while the probe is in and cooling the cochlea, right? So this is to kind of illustrate that it doesn't come in this, doesn't impede the surgeon by doing in the cochlear implant surgery. And this is actually a little cochlear implant that has been inserted through the round window into the cochlea, right? This is a human temporal bone study. We took like 10 different temporal bones. We did micro CT scanning of them. We looked at all the parameters that a surgeon would use in their practice as they're thinking about what their approach is going to be. So are they going to make a facial recess? Are they going to need to expand the facial recess so that they can view the entire round window? Are they going to think about, all right, in this particular case, we won't be able to see the round window, so do we need to do cochleostomy, right? So all of these different things that we wanted to look at, we looked at, so for example, some of the things that just I highlight here, so facial recess area. So that's that little area, the cavity that a surgeon drills out to be able to see the basal turn of the cochlea, the round window, and then insert the electrode, right? So that's where our probe's going to go to cool the cochlea. So we want to know, is that a restrictive factor in our hypothermia application? And you can see that there's a very wide range of facial recesses that we had. The angle where the ear canal comes in, right, and it makes, where is the facial nerve located? We don't want to cause damage to the facial nerve or anything like that while we do that. So we can look at many of these features. And then what we did was we asked these surgeons to actually go in and drill out the facial recess, put in the probe, and evaluate. We asked 3 different neurologists in our department to evaluate, does the probe impede your view of the round window? Right? And essentially all of them give a category of like, 1 to 1. This is called a St. Thomas Hospital classification that I won't go big in details into, but essentially what this tells us that, they could still view the round window 100%, right? So that's, again, things that you want in a translational application, right? And then we had actually the human temporal bones and we cooled them down and we see whether can we actually cool the cochlea. So we took the whole bones, we warmed them up to 37 degrees. We maintained them in a hot bath so that they stay at 37 degrees. We put in our probe, right? And then we start cooling, and we see, like, using our protocol that we used in animals, can we get the cochlea down to about 33 degrees? And we can, right? So that's again, like, really nice verification that this will work in humans, right? When we go in. So the next step, of course, is to actually go into patients and see whether we can translate this into the OR. So we're working with a company called Cochlear, which is the largest manufacturer of the cochlear implant, and building these devices and trying to think about, like, can we actually move this into patients. So I'll switch gears a little bit and talk about something slightly different, something that you may have heard about, but maybe not in detail, just like cochlear implants, called noise-induced hearing loss. I don't know if you guys, are on NPR or things like that all the time, but this has received a lot of publicity in the recent years, and I'll try to tell you a little bit about why that is. So I have a couple of sounds, okay? So try and hear the sound that's being played, and there's going to be a sentence in the middle at some point. See if you can decipher what that is. Did you guys catch that sentence? What was it? Okay, so he has some filters there, which made things a little bit easier, but if I...
It's always great to be a Miami Hurricane.
So if I change the ratio of that background noise to the speech sentence, right, you can hear it even better, right? So from the first time to the second time, you probably noticed a big difference, right? So that's what a normal cochlea does, right? So it takes all the frequency information that it has, and it thinks about what the, why is there an echo? Anyways. It thinks about figures out what's the background noise, what do I need to suppress, and then what's the speech that I need to bump up, right? So that's what the natural cochlea is doing all the time. But as we age, as we lose those outer hair cells, we start losing some of those inner hair cells. What happens is this cocktail party effect, right? So when we were at a restaurant, a loud restaurant or something like that, and there's a big group, many times, at least at my age, I have to look at the person, right? I have to look at the face of the person, and then that helps me sort of focus my attention, right? So I have to use a little bit more of an effort to try and hear what they're trying to say. And this is a big deal. So this is, age-related changes that are happening. This is studies coming out of Harvard Mass Eye and Ear Infirmary, and what they're showing is in humans and in mouse, what you're seeing is cell survival over age, right? So by the time you get to about 40, the hair cell survival, the neuron survival, all of that, the outer hair cells, inner hair cells, as well as the neurons, the auditory nerve fibers, right, start to go down quite fast, right? So you're losing a lot of those, that resolution that we had in terms of frequency specifications, right? Now, add to that noise exposure. Right? So that was the natural aging process that's happening, right? Where after 40 years of age, we sort of start losing some of those outer hair cells, some of the auditory nerve fibers, some of the synapses to the inner hair cells, et cetera. This is a study by Sharon at Mass Eye and Ear in 2009 that actually sort of kick-started some of these things. What they did was they took mice, right? These are wild-type mice. And what they did was they looked at the inner hair cells. So these are the nuclei of the inner hair cells. They are the ones that communicate sound information to the brain. And they looked at the number of synapses that these guys have. So this is one hair cell that they outline. And these reds are the number of synapses that a spiral ganglion or auditory nerve fiber makes on that hair cell, right? And what they did was they had another group of mice that they gave noise to. A noise that's equivalent of going to one small rock concert, right? 2 hours, 95 decibels. That's all it is, okay? Actually, rock concert's about 105, so this is actually a little bit below rock concert too. And what they found is if you just look one day after, the number of synapses are significantly down, right? So the number of synapses that the auditory nerve fiber makes on the inner hair cells are significantly down. Then what they did was they looked at the mice 2 weeks post-exposure and 64 weeks post-exposure, right? So now they're adding in that aging effect that might be happening, right, into it. And what they found was that the ganglion cells, so now the cell bodies of those neurons, which are downstream, right, into the cochlear nerve, are getting damaged significantly higher rates. So you take, for example, your age-related hearing, and you add noise to it. So this red is 100 decibel noise, one time added, and what you see is that there is an exaggerated loss, right, at different frequencies of the spiral ganglion. So now, the people who are going to, and I don't mean to scare anybody here, but the people who are going to rock concerts, people who are going to Ultra, right, they become customers of our department much earlier. Right? Which is, I guess, if you're a neurologist, that's a good thing. If you're a cochlear implant company, that's a good thing. But if you're a patient, that's a really bad thing. So, if you look at CDC data, 24% of adults, 17% of teenagers, and I think this is underestimated, show hearing loss from exposure to loud sounds. Right? That's a scary statistic. 40 to 50 million people have trouble with their hearing because of noise exposures, right? And they actually predicted that you could save $123 billion in healthcare costs if only we could reduce 20% of hearing loss from noise exposure. Now, this also includes military population where blasts are going on all the time, they're at a gun range, practicing 20 days in a straight row when they're training to get ready for their service. They're training in and going with their guns blazing. So there's a significant amount of loss. And so, again, we know, in my lab, I know a lot about hearing. I know a lot about animal models. Now I know a little bit about hypothermia. So we said, can we cool the cochlea after noise exposure? And would that do anything, right? So what we did was we created this little sort of neck brace. So think about this as your airplane pillow that you might have, right? That's just cooled, right? So you put it on the back of the rat. And then what we do is we give the rats, one group of rats, noise exposure, 95 decibels, so much lower noise exposure, for 2 hours. And then after 15 minutes, we do a hypothermia. So we give them the neck brace and we cool their cochlea down, and we can actually, this is non-invasive, so this is just from the neck brace. So we're not doing surgical insertion of anything, right? So we're just doing non-invasively. And amazingly, we could actually cool the cochlea just through the neck brace by about 5 degrees, right? So we said, all right, can we try this and see what happens in rats? So we went ahead and we looked at what happens to normothermic rats who get noise exposure. So just look at the red curve, for example. This is day 1 after noise exposure. And this is, again, their hearing threshold. So there's about a 40 decibel change. This is day 1 in hypothermic animals, right? So you're getting significant protection on day 1. At day 28, you get this notch, right? This gray bar is where the frequency of noise damages, and you see that the frequency is 1 octave above. Even at day 28, there's a certain amount of hearing loss that we have, whereas in the hypothermic animals, we've kind of recovered pretty much all of the hearing, right? So good thing so far. We looked a little bit more closely at how the two sets of animals do, and again we see this significant recovery with hypothermia at day 1, lasting up till day 28, and we have done up till day 84 now. We can look at those little structures, those synapses that I was telling you about, right? And we can look at that in 2 ways. We can look at the amplitude of that wave 1 of the ABR that I told you about, which is the cochlear nerve response, right? And we can measure the amplitude, and we can create growth curves of that at different sound levels. The black line is before either group had noise exposure. The blue lines are over time of hypothermia group, and red lines are the normothermic group, right? So you see that the red lines always continue to still be down, right? Even at day 28. They're still not recovered as much as the blue line has. And if you look at the synapses themselves, right, what we find is this is a wild type, right? So this is a controlled cochlea, no exposure. This is a noise-exposed cochlea, and this is a hypothermic cochlea. And what you might appreciate is that there's a lot more red dots here compared to over here, right? So we're just with that, again, one-time application of hypothermia, we're preserving a lot of those synapses intact, right? Again, a good thing, I think. We did I'm big time into this RNA sequencing data now, just because I think the plots look really, really cool, right? So we made some heat maps of animals that were getting noise or getting hypothermia. And so this is, again, controlled cochleas are C's, normothermic noise exposed cochlea are N's, and hypothermic noise exposed cochleas are this. And you can see that the hypothermic look actually just like normal cochlea, right? Versus the normothermic have quite reversed gene expressions. So this is just fresh data that came out actually just this week. So we're still analyzing what are those gene pathways that are getting affected. Again, we see the same things. The Cas species are significantly down. TNFs are down, IL-1 beta, and the CCLs are kind of significantly getting down. So we know, again, we're doing the right things to the cochlea, which is, again, good news. So what we're thinking about is that, maybe there is potential application of this non-invasive airplane bag, as you will, like a neck brace for patients who might want to still go to rock concerts or football games. I've actually recorded noise level exposures at several places now. So if I took my daughter, who is a three-year-old, to Disney on Ice. If you guys can imagine, the Disney on Ice show has 117 decibel level noise at times, right? So you have thousands of little kids going into this Disney musical, right, getting exposed to noise, and who knows what it's doing to their ears 20 years down the line, right? You go to a Miami football game. Again, I love the Canes and I want to support them. But when they start playing different musics over there, the sound reaches 107, 108 decibels, right? So now we're exposing all these people to different amounts of noise. If you think about, like, Seattle Seahawks, right? So they pride themselves in the 12th man, right? And they reach sound levels about 130 decibels when they're cheering loud, right? That's damaging noise that we're exposing the world to, right? So we think that there's applications of this to try and help, again, like, patient groups. And again, one of the things, the reasons why drugs have had a very difficult time in this particular case is that, one, cochlea is a very protective structure. It's very difficult to get drugs into the cochlea. And number two, like, systemic applications that people have used of different drugs, what they require is 2 weeks before you're going to get noise exposed, you're going to have to start taking these drugs. Well, good luck trying to get anybody to do that, right? Anybody who wants to go to ultra, they're not going to take these medicines 2 weeks in advance every single day for 14 days just to be able to go to ultra, right? So that's one of the reasons why I think, like, the drugs haven't translated. So I think, like, non-pharmaceutical approaches like this have significant advantage. And one of the things that, with John Barrett and others here that we're looking at is, can we repurpose the drugs in a different way? So can we now think about a combination, this hypothermia applied right after noise injury or cochlear implant injury or others, can allow us to expand that critical time window when we can come in with complementary drugs, right? So now it's all after the trauma. It's not before the trauma that you have to do. And maybe these drugs will now have an added benefit to what we're doing, right? And so this is just some in vitro data kind of showing you that if you do oxidative stress in different cells, the cell viability goes significantly down, right? Hypothermia will preserve some of that. The drugs really don't make a lot of difference. These are, like, 3 different antioxidants. But if you combine it with hypothermia, all of them have synergistic effects, right? So they significantly increase cell viability. So this might be, like, one of the ways in which we can actually think about how we can redo all of the studies that have been done in terms of drugs, use that information, but with a combination therapy, like with hypothermia, that's delivered non-invasively. So again, like, we're looking at the spiral ganglion loss. So I told you that, we looked at that functional loss. We can protect that up till 84 days, even for the noise cases. We looked at, like, the synapses, and we're preserving a lot more of the synapses just with hypothermia, which is, again, good news. What eventually does happen to the spiral ganglion, right? So maybe next year I'll come in and tell you guys about what happened to the spiral ganglion, like, and especially when we combine that with aging, right? What's happening? Are we preserving the nerves much more with applications like this? We don't know yet, but the experiments are underway, so stay tuned. So anyways, just to kind of wrap it up, this is my lab group, a really great group of people, students and postdocs doing really amazing work. Collaborators over here, including John and others, Jay Lee, who's not listed here, I don't know why, and then some of the funding support that my lab has. So anyways, thank you, and any questions that you guys might have about hearing or hearing loss, I'm happy to answer.
Thank you for sharing this.
Yeah, good.
So, the implications are enormous.
Yeah.
Don't let the trial lawyers hear about this, because think about, we're working on a new tower above the operating room. So all day, this is
Yeah.
staff, but it's so many.
Absolutely. I think, and like, one of the things that, now that I got into the noise-induced hearing loss portion of the work only recently. So this is about a year old, right? Data that I have, year old that I started working on that. But since then, I've become hyper aware of noise that's around us, right? And one of the things that I do is I look at, if you look at people that are working on jackhammers, for example, at construction sites, people who are using pressure washers to clean like sidewalks or something like that, one of the things that you realize is that none of them have their hearing protections inside. And if they do, what they do is they'll have one ear, but the other ear uncovered so that they can still hear the surrounding noises or talk to their friends or something like that, right? And I can't even imagine, like, and I have all the measurements, like, because I have a Decibel Pro app on my phone to measure, like, how much sound decibel it is. So that's what I do. Like, every time I hear a noise, I'm just like, let's measure, like, what that sound is. And it's always surprising. It's always hundreds of decibels, right, that we're exposing our ears to.
What is the damage level?
So that's like, Sharon Kujawa and Charlie Lieberman at Massa and Ear, what they've shown is that you can go as low as 85 to 90 decibels, which is quite low, right? So when we talk loudly, we're going to 85 decibels. So beyond that, if you're going where you're hearing like, music when you go to restaurants, for example, or the loud restaurant noise that you have, that's getting to about 90 decibels. And what they've shown in mice is that if you do one time with that noise, you may not see a significant effect until you cross a threshold, but when you expose it sort of daily over time, you start getting accumulated damage. You had a question, sorry. Yeah, so that's challenging, right? One of the reasons why it's challenging is because it's very difficult to probe a human's ear. And the other challenge is that the measurements that we have right now, right, in terms of the auditory brain responses, for example, DPOE, so distortion product acoustic emissions that you can measure back from the outer hair cells, they're all still limited in that they don't tell us specifically this loss of synapses, right? So we can't really probe that, particularly in humans. There's a lot of data now coming out of a couple of labs, particularly this Massa and Ear, because they have this huge temporal bone registry. And one of the things they've done working with NIDCD is they have started to kind of look at these human temporal bones. And one of the things that they're trying to do is go back into their files, the records, and figure out like how much noise exposure they've had over time, for example, and correlate that with the loss of spiral ganglion, the loss of the synapses, the loss of auditory nerve fibers. But as I showed, like, that one data set that we have where they showed that when you add the noise to just the aging, right, you start getting that very steep decline in auditory nerve fibers, right, and the synapses. They're starting to come out with more data about that. We still don't know. We don't have the final answers yet, but it seems like, at least from all the indications, that there is a significant additive effect of noise also happening in patients. And this has implications not just, you're right in one way that mice have much more refined hearing, so their frequency band is much higher. So they'll go from 200 hertz to about 50 kilohertz. We go only from about 200 hertz to about 20 kilohertz. And 20 kilohertz is about a 15-year-old pristine ear, right? All of us in this room are 16 kilohertz or lower, right? So we can't hear 16 to 20 kilohertz. Maybe one or two of you might be able to. But even as we lose that hearing at the higher frequencies, it has implications about noise in speech and noise. So sort of like the sentence that I played when we're going to restaurants and trying to hear like somebody rather than just one-on-one interaction. So it has a significant interactions from there. So there is anecdotal evidence coming in from all of that. There's a lot of data coming in now from VA study. So VA has a similar plan I don't know if you guys knew about this, but like, you have the All of Us program, right? That's funded through NIH, where they're collecting like, million data sets from people, including genome data, including all of their history, et cetera. VA has a similar plan called Million Veterans Program. And what they're trying to do is get million veteran data, including some of these factors now. So I was at a meeting led by VA, and one of the things as a working group we suggested is that they need to start collecting noise exposure data from these patients, right? So as we get their genetic data and as we get some of this information that we need from like, a couple of million people, we'll start to have some of those answers. But, yeah, it's a long road, but exciting road. Yeah. All right. So anyways, my lab is on the 4th floor, so if you guys ever want to stop by and talk about hypothermia or hearing or whatever, just let me know. All right, thanks.